Why did John B. Fenn, United States, and Koichi Tanaka, Japan, win the Nobel Prize for Chemistry?
For the development of methods for identification and structure analyses of biological macromolecules. For their development of soft desorption ionization methods for mass spectrometric analyses of biological macromolecules and Kurt Wüthrich. Switzerland, for the development of methods for identification and structure analyses of biological macromolecules for his development of nuclear magnetic resonance. Spectroscopy for determining the three-dimensional structure of biological macromolecules in solution. In the past, mass spectrometry was limited to the study of relatively small or lightweight molecules. Fenn and Tanaka share half of this prize for their work in extending the technique of mass spectrometry to significantly larger biomolecules. Fenn and Takano independently developed two different methods for obtaining freely hovering protein samples appropriate for mass spectrometric analysis. Wüthrich received the other half of this prize for the use of NMR spectroscopy to determine the three-dimensional structure of biomolecules in solution. This represents a significant advance beyond crystal structure analyses, as the structure of biomolecules may differ in the crystalline and solution phases, with the solution phase structure being more relevant to their true biological function.
What is the signal-to-noise ratio in a measurement?
The signal-to-noise ratio is a comparison between the strength of a measured signal to the background variation naturally present in the experiment. There are different ways of reporting the signal-to-noise ratio, but one commonly used measure is to compare the mean value of a measurement to the standard deviation of the background region. For example, we might want to measure the intensity of a laser beam using a detector. The signal would be the measured intensity when we shine the laser beam on the detector. The noise would be measured as the standard deviation of the intensity measured, due to the ambient light when the laser was not shined onto the detector. Such a definition is commonly used when looking at weak signals in imaging or microscopy. Choosing an appropriate way of reporting the signal-to-noise ratio can depend on details of the situation in question, such as whether the data can take on both positive and negative values, or whether the data can only have positive values.
What is insoluble?
A substance that does not dissolve in a solvent.
What is a CD made of?
All types of optical discs, CDs, DVDs, Blu-ray, discs, etc., have basically the same components. The outer layer of clear polycarbonate plastic protects the inner layers that contain the data from damage. A layer of a highly reflective metal, usually aluminum, is used to reflect the laser that is used to read the data. The data itself is stored on another polycarbonate layer that contains teeny tiny little pits. The pits are arranged in spiral tracks, just like a vinyl record. If you've ever seen one of those, and are about 100 nanometers deep and about 500 nanometers wide. The laser can detect the change in height by measuring changes in how the light is reflected.
What is an allotrope?
Different arrangements of atoms of a single element, e.g., diamond and graphite are allotropes of carbon.
What does enantiomeric excess measure?
The enantiomeric excess is a measure of how much more of one enantiomer is present in a mixture. It's often reported as a percentage. Racemic mixtures have an enantiomeric excess value of 0% because both enantiomers are present in equal amounts. For a solution composed of 75% of one enantiomer, the enantiomeric excess would be 50%, 75% 25% equals 50%.
Leloir, Argentina, win the Nobel Prize for Chemistry?
For his discovery of sugar nucleotides and their role in the biosynthesis of carbohydrates. Leloir discovered a substance essential for the transformation of one sugar into another. And this turned out to be a sugar bound to a nucleotide molecule, a sugar nucleotide. He soon realized that he had opened the door to a vast number of unsolved problems surrounding carbohydrate synthesis, and proceeded to pursue them with fervor. During his career, Leloir revolutionized the understanding of the synthesis and biosynthesis of sugars.
What chemicals are typically analyzed in a urine analysis?
Just like a blood analysis, there are several analytes that can be looked at by urine analysis. pH, density, proteins, glucose, ketones, leukocytes, blood, or human chorionic gonadotropin, the presence of which can indicate pregnancy. The results can indicate several things about a person's health. If the person is well hydrated, for example, a lower density would be expected. The presence of proteins in the urine is uncommon, and this may indicate that the patient's kidneys are not functioning properly. Similarly, glucose and ketones should not be present in the urine. But if they are, their presence could be a symptom of diabetes.
How is nuclear chemistry related to the alchemist's goal of transmutation?
Alchemists sought a way to turn common metals into gold, which we now know is not possible to do in any simple way. The reason is that transmutation would involve converting one element into another, which can't be done by simple chemical processes. It would require a nuclear reaction to take place. Either a heavy nucleus would have to divide into a gold nucleus and another byproduct, or two lighter nuclei would have to combine to form one of gold. Neither of these things happen readily. If early alchemists had recognized the distinction between more ordinary chemical reactions and the nuclear reaction they were looking for, it would likely have saved a lot of time and effort.
What pollutants do nuclear power plants generate?
Not many, unless something goes wrong. No greenhouse gases, like carbon dioxide, are generated in nuclear power plants. And each only produces roughly 1 cubic meter of waste per year. As long as this is properly taken care of, nuclear power plants are one of the cleanest forms of energy.
What is aqua regia and what is it used for?
Aqua regia translates to royal water in Latin, and it consists of a mixture of concentrated nitric and hydrochloric acids, typically in a 1:3 ratio, respectively. Since these are both strong acids, aqua regia can be quite dangerous and must be handled with extreme caution. This mixture is capable of dissolving several types of metals and has found many applications in chemistry, including the refinement of very high purity gold.
What is dry ice, and why does it evaporate from the solid phase?
Dry ice is solid carbon dioxide, CO2. At constant atmospheric pressure we see that an increase in temperature will bring it from the solid phase directly to the gas phase. This process is called sublimation, and it takes place at -78.5 degrees Celsius.
What is the hardest material in your body?
Tooth enamel is the hardest material in your body. It's even harder than bone.
How do mirrors reflect light so well?
Most modern mirrors are made of smooth layers of silver or aluminum, along with other chemicals and coatings to aid in their construction. while the chemical properties of silver play some role in reflecting light. The fact that these layers are very, very smooth is more important here. If the surface of the silver layer were rough, the light would be reflected in a variety of different angles, which you would see as a distorted image. When the surface is perfectly smooth, the light bounces straight back into your eye and you see an accurate reflection of the object. This is why you can see clear reflections in very still water or a shiny piece of leather. Both are smooth surfaces.
Who first realized that air has weight?
It was actually a mathematician named Evangelista Torricelli who is the first on record to demonstrate that air has weight. His experiment to prove this fact was prompted by the observation that water from a mineshaft could only be pumped upward to reach a certain height. Torricelli thought that the air pushing down on the surface of the water must play a role. To test this theory, in 1643 he placed a sealed tube of mercury upside down in a bowl of mercury. He observed that the weight of the air would keep the mercury in the tube at a certain level. And on different days he observed that the mercury would rise to different levels. We now know this is because the air pressure varies from day to day. And Torricelli's experiment was the first barometer.
What makes a reaction happen spontaneously?
A spontaneous chemical reaction is one for which the associated change in Gibbs free energy is negative. The fact that a reaction is spontaneous actually doesn't tell us anything about how quickly the reaction takes place, though. A spontaneous reaction may happen very quickly or take thousands of years.
How can I observe layers of immiscible liquids?
Chemistry principles encountered in this experiment: density, miscibility, polarity, materials. You'll need asterisk. Honey, pancake syrup, liquid dish soap, water, vegetable or cooking oil, rubbing. Alcohol, lamp oil, a tall glass of water or other container, optional, food coloring to improve visibility asterisk note. For this experiment, it is not necessary to have every material listed. The procedure: pour the densest liquid into the glass first. Note that the liquids above are listed from most dense to least dense. Try to avoid letting the liquid run down the sides of the glass. Gently pour the second liquid on top of the first. One way to pour it a little more slowly is to pour the liquid over another object such as a butter knife or the backside of a spoon. Allow each layer of liquid to settle for at least a few seconds before adding the next liquid. You'll notice that, instead of mixing, the liquids tend to stay in separate layers. The reason for this is that they are immiscible, which means that it is more thermodynamically favorable for the liquids to stay separated in layers and to form an interface than it is for them to mix together. Whether or not two liquids will be miscible is dictated by the details of the entropic and enthalpic factors associated with the mixing or separation of the two liquids in question. This is often directly related to whether or not the compounds have similar polarity. For example, we know water is a very polar substance, while vegetable oil is composed of primarily long, nonpolar hydrocarbon chains. These do not interact favorably with one another, and prefer to stay in separate layers. Continue pouring the third, fourth, etc. liquids on top of each other in order of decreasing density. You can just follow order of your liquids in the list above. As you pour in the successive liquids, they should continue to form separate layers. The densest liquids are affected the most by gravity, so these tend to stay below the less dense liquids. These liquids are not miscible, so they do not mix together to form a single solution. In truth, if you wait long enough, some of these liquids will mix together. But it will take a while. That's it. You should now see a series of separate liquid layers in your container.
How can microwaves be used to promote green chemistry?
Microwaves are electromagnetic radiation in the frequency range of 0.3 to 300 GHz. When microwaves are absorbed by many substances, it causes their temperature to increase, thus heating a sample. This is also the same way the microwave in your kitchen heats and cooks food. Microwaves thus offer chemists the opportunity to use heat to promote reactions in cases where conventional heating methods are not possible. This can be useful for promoting reactions under green conditions, such as when we desire to heat the reagents involved in a reaction in the absence of a solvent.
Do chemists really study food chemistry?
Yes, they really do. There's even a scientific journal called Food Chemistry, published by Elsevier, dedicated to reporting new findings regarding the chemistry and biochemistry of food and raw food materials.
Why did Osamu Shimomura, Japan, Martin Chalfie, United States, and Roger Y. Tsien, United States, win the Nobel Prize for Chemistry?
For the discovery and development of the green fluorescent protein, GFP. These three scientists received the prize for their work with a protein simply called green fluorescent protein, or GFP. This protein was first observed way back in 1962 in a jellyfish. It eventually became a very important molecule in the biological sciences, as clever researchers have found ways to use GFP to study otherwise invisible chemical processes. Shimomura, Chalfie, and Xian were at the forefront of the major discoveries that led to the understanding of GFP that researchers possess today.
What is a levorotatory?
Having the property of rotating plane polarized light counterclockwise.
Does metal rust in outer space?
Sort of. Rusting in outer space doesn't happen in quite the same way it does on Earth due to differences in the amount of available water. On Earth, iron rusts when it interacts with water molecules, causing oxidation of some of the metal atoms to metal oxides. Recalling this information, it is clear that some source of oxygen atoms must be present for metal to rust. There is very little oxygen or water floating around in outer space. So the reaction doesn't proceed as quickly or via the same mechanism. Actually, in outer space, the very small amounts of oxygen, O2, or water, H2O, that are around are believed to undergo photochemical reactions with metals to produce metal oxides, like rust, Fe2O3. Scientists can get a sense of how rapidly metals rust in outer space by looking at iron-containing meteorites that reach the Earth.
What is DDT?
DDT, or dichlorodiphenyl trichloroethane, is a substance that was widely used as an insecticide until its harmful effects on human health and on wildlife became known. DDT was essentially poisoning the humans and wildlife who came into contact with it. In the context of the advent of green chemistry, DDT carries a special significance in that it helped to awaken the public to the fact that the indiscriminate use of chemicals was causing harm to the environment. News of the harmful effects of DDT was spread by Rachel Carson's 1962 book, Silent Spring, which explained the numerous negative environmental effects of spraying DDT on crops. Knowledge of the harmful effects of DDT helped to awaken the public to the idea that releasing large amounts of relatively untested chemicals into the environment was potentially causing damage to humans and wildlife. DDT was officially banned in the US in 1972.
How was oxygen gas first discovered?
Well, to answer that question, you would first want to know who first discovered oxygen. And there is no simple answer to that question. There are three people to whom discovery of this can be ascribed: Carl Wilhelm Scheele, Joseph Priestley, and Antoine Lavoisier. Scheele produced O2, he called it fire-air, from mercuric oxide, HgO. In 1772, but the result wasn't published until 1777. Meanwhile, in 1774 Priestley produced O2, he called it dephlogisticated air. Using a similar experiment, which was published in 1775. Lavoisier claimed to have independently discovered the gas, and was in fact the first to explain how combustion worked via quantitative experiments, leading to the principle of conservation of mass, and ultimately disproving the entire idea of phlogiston. WIO. So Scheele found it first, but didn't report it. Priestley reported it first, but didn't have the explanation correct, and Lavoisier was last, but nailed it? Who would you give credit to?
What is the melting point?
Temperature at which liquid and solid phases coexist in equilibrium.
How does your liver process alcohol?
The liver contains an enzyme called alcohol dehydrogenase, which is responsible for metabolizing ethanol. Alcohol dehydrogenase converts the ethanol to another molecule called acetaldehyde, which is then excreted from the body. A healthy liver can process about half an ounce of pure ethanol each hour, which equates to roughly 1 beer, 1 glass of wine, or an ounce of liquor each hour.
What is a barometer?
An instrument used to measure pressure.
What is a tautomer?
Structural isomers that differ only in the position hydrogen atoms.
What is volume?
The quantity of space that a substance occupies.
What is physical chemistry?
Physical chemistry is a branch of chemistry primarily concerned with developing a better understanding of the fundamental principles that govern chemical processes. It is an empirical science, meaning that it is based on experimental observations. Though it is probably the most closely linked experimental branch of chemistry to developing new theories in chemistry. As the name implies, physical chemistry is intrinsically concerned with topics in physics that are also relevant to the study of chemistry.
What are some examples of biological feedstocks?
it is desirable to use biomass, or plant-based materials, as feedstocks for chemical synthesis and energy production. Through photosynthesis, plants are able to efficiently capture and store energy from sunlight. And finding ways to use biomass for green chemistry applications is extremely advantageous in advancing the goals of the field. Sources of biomass can be grouped into several categories, including cellulose, lipids, lignin, terpenes, and proteins. Cellulose is often found in structural parts of plants. Lignin is a polymer often found along with cellulose in wood-like parts of plants. Lipids and lipid oils are often extracted from seeds and soybeans. Terpenes are found in pine trees, rubber trees, and a selection of other plants as well. Proteins are found in relatively small quantities in many types of plants and also in larger quantities in animals. Some efforts are also underway to use genetic transplants to create plants that produce increased amounts of proteins. One of the primary challenges in using biological feedstocks to produce chemicals or energy involves separation and purification of the desired materials.
What is a geometric isomer?
A molecule with the same molecular formula but different arrangement of those atoms in space.
What is a meniscus, and how should it be read?
A meniscus is the curve in the surface of a liquid contained within a usually narrow container, such as a graduated cylinder. Take a look at the graphic to the right. The numerical value we read for the volume of liquid contained in this cylinder will depend on what part of the meniscus we choose to look at. Graduated cylinders and other containers are calibrated such that we should get the most accurate reading by looking at the center of the meniscus from eye level. If we don't look at the meniscus from eye level, something called parallax error will occur that will distort our perception of level of the meniscus.
Why did Johann Diefenhofer, Robert Huber, and Hartmut Michel, all Federal Republic of Germany, win the Nobel Prize for Chemistry?
For their determination of the three-dimensional structure of a photosynthetic reaction center. This prize was awarded for the atom-by-atom characterization of a membrane-bound protein. And, in particular, one that is responsible for carrying out photosynthesis. These proteins are quite difficult to crystallize, and Michel can be credited with achieving this task. He then collaborated with Huber and Diefenhofer to unravel the details surrounding the structure of the crystallized membrane-bound photosynthetic reaction center.
How is the rate of a reaction affected by temperature?
The rate of a chemical reaction will generally increase with increasing temperature. This is because a higher temperature translates into a higher average energy per molecule, which makes it easier for molecules to surmount the energetic barrier to the reaction. In terms of how this fits into the rate equation, the rate constant K depends on temperature. And K almost always, but there are exceptions, increases with increasing temperature.
How many amino acids are found in humans?
There are 20 amino acids commonly found in humans. They are classified into 4 groups based on the identity of their side chain. The 4 groups are polar, nonpolar, acidic, and basic. These 20 amino acids are what make up all of the proteins and enzymes in your body. The interactions between the individual amino acids determine the overall conformation of the protein. Though the relationship between the sequence of amino acids and the structure adopted by the protein is very difficult to predict.
Why did Adolf Otto Reinhold Windaus, Germany, win the Nobel Prize for Chemistry?
For his research into the constitution of the sterols and their connection with the vitamins. Windaus was awarded the Nobel Prize for discovering that cholesterol, a sterol, was removed to cholecalciferol, vitamin D3, through a series of several steps. One of Windaus' doctoral students, Adolf Butenandt, was himself a Nobel laureate.
What properties influence solubility?
The most significant properties are intermolecular forces and temperature. If there are favorable interactions between the solute and the solvent, solubility will be higher. This is actually a balance of the interactions of the solute with the solvent and the stability of the solute in the solid phase. Temperature also influences solubility, and for most substances, solubility increases as the temperature of the solvent rises.
What is spectrophotometry?
Spectrophotometry is a technique that involves passing light through a sample to measure the fraction of light transmitted or reflected by the sample. This can be useful for determining the concentration of an analyte in a solution or for determining the identity of an analyte present. A law known as Beer's law, see also physical and theoretical chemistry, is particularly important in spectrophotometry. Beer's law tells us that the concentration of a species is directly proportional to its absorbance in a solution. This means that if we know the absorbance of a solution of our analyte at a known concentration, we can determine the concentration of that analyte in any other solution.
What is the scientific notation?
A way of expressing a number is the product of a decimal and a power of 10, for example, 1050 equals 1.05 x 103.
How do space probes, like the Curiosity rover, look for molecules on the Moon or Mars?
The Curiosity rover has a whole suite of chemistry tools on board. The laser-induced breakdown spectroscopy (LIBS) tool is probably the coolest. This instrument breaks down rocks and bits of soil by firing a freaking laser at the target. The elements that made up that rock are then detected by atomic emission spectroscopy. Curiosity also contains an alpha particle, He2+ ion, X-ray spectrometer. APXS, which is also used to measure what elements make up a sample. If the NASA scientists want to know more than just what elements make up a sample, they can use the quadruple mass spectrometer, which can measure the mass of ions of gases and organic compounds.
What is a bond angle?
The angle relating the orientation of two bonds connecting three atoms.
What is an atom?
Atoms are among the most basic building blocks, making up all matter. The word atom derives from the Greek word atomus, which means that which cannot be split. The existence of atoms, or a fundamental, indivisible unit of matter, was proposed long before modern chemistry and physics came about. It turns out that atoms are actually made up of even smaller particles. But the atom is the smallest unit of matter that defines an element. The smaller particles that make up an atom are positively charged protons, charge-neutral neutrons, and negatively charged particles called electrons.
What is the collision theory?
Defines reaction rates as a function of the collision frequency.
What is a kilogram?
Standard base unit of mass.
How does the density of snow compare with that of liquid water?
The density of recently fallen, damp snow is approximately 1/10 that of liquid water. This means that 1 centimeter of rainfall and 10 centimeters of snowfall contain approximately the same quantity of water molecules.
What is bioremediation?
Bioremediation is an approach to removing pollution from the environment that relies on the use of microorganisms to metabolize pollutants into non-toxic products. Sometimes these microorganisms have been genetically engineered for a specific application. For example, a bacterium called Deinococcus radiodurans has been genetically engineered to digest ionic mercury compounds and toluene from nuclear waste sites. In such cases, bioremediation can often be accomplished by introducing a microorganism at the site of the pollution, thus avoiding the need for physical cleanup and transportation of the waste to a new location.
What is Dalton's law?
A law regarding partial pressures of gases that states the total pressure of a mixture of gases is equal to the sum of the partial pressures of each individual component.
What molecular properties lead to higher boiling points?
There are a few factors that play important roles in boiling points of substances. The first is molecular weight. In general, heavier molecules have higher boiling points, which is pretty straightforward considering how we defined boiling point above. Heavier molecules take more energy to move from the liquid to the gas phase. All other properties that affect boiling point deal with intermolecular forces, or interactions between molecules. Think of this like the affinity of one molecule of a substance to be attracted to, or stick to, another molecule. Non-covalent bonds, like ionic or hydrogen bonds, significantly increase boiling point. Why? Because to move to the vapor phase the molecules typically have to break these interactions. Dipole interactions and van der Waals forces have similar effects, see atoms and molecules. But these interactions are weaker, so the effect they have on boiling points is smaller. Finally, branching of the carbon backbone of a molecule is also frequently touted as a factor leading to lower boiling points. While this is true, it is really the weakening of van der Waals forces at work here too.
What happens when you add sodium metal to water?
Sodium metal reacting with water can produce a violent response. Sodium and water react according to the equation, 2Na, plus 2H2O, L. 2NaOH, Aq, plus H2, G, plus heat. As you can see, this reaction produces heat. and it turns out that the amount of heat produced can be pretty substantial. The heat can even cause the hydrogen gas, produced in the reaction, to ignite. Thus reacting according to this equation, H2, G, plus O2, G, H2O, G. Plus heat. The heat produced by both of these reactions can even cause any currently unreacted sodium metal to also ignite and burn according to the equation. Na, S, plus O2, G, Na2O2, S, from these reactions. It is probably clear that the reaction of sodium with water can produce quite a lot of heat.
How much space does the nucleus take up?
The nucleus of an atom takes up a very, very small fraction of the total space occupied by the atom. The diameter of a nucleus is on the order of 100,000 times smaller than that of a whole atom.
What is molality?
A measure of concentration defined as moles of solute per kilogram of solvent.
What is an isotope?
Atoms of the same element, same number of protons, with different number of neutrons.
What is the boiling point elevation?
A colligative property, the increase in boiling point of a liquid as a solute is added.
What is the auto-ignition temperature of a substance?
The auto-ignition temperature is similar to the flash point, except that it does not require an ignition source to begin or to continue burning. Above the auto-ignition temperature, the vapor will start to burn, and continue to burn, even in the absence of any ignition source.
What is an electrophile?
Electrophiles are species that accept electrons, from a nucleophile, in a chemical reaction. Usually, electrophiles have either a full or partial positive charge, or in some cases, like BH3, have an unfilled octet of electrons.
Has any legislation been passed regarding the implementation of green chemistry?
Also yes. A couple of examples are the registration, evaluation, authorization, and restriction of chemicals. REACH, program in Europe and the California Green Chemistry Initiative in California in the United States. The purpose of the REACH program is to require that companies make data available that demonstrates the safety of their products. This includes the potential chemical hazards during the use of a product. And it also describes means of restricting the use of specific chemicals. A similar piece of legislation, the Toxic Substances Control Act, exists in the United States, but this has received criticism for being far less effective. The California Green Chemistry Initiative was approved in 2008, requiring the California Department of Toxic Substances Control to place priority on specific chemicals of concern.
Why did Harold Clayton Urey, United States, win the Nobel Prize for Chemistry?
For his discovery of heavy hydrogen. Urey received the Nobel Prize for isolation of deuterium D2, by distilling a sample of liquid hydrogen multiple times. He is perhaps better known, amazing that he's better known for something other than what he received the Nobel Prize for his work on the Manhattan Project. Working at Columbia, Urey and his team developed a method for enriching uranium by gaseous diffusion. Finally, after World War II ended, at the University of Chicago, Urey and Stanley Miller, one of his graduate students, showed that a mixture of water, ammonia, methane, and hydrogen could produce amino acids by exposing the mixture to electricity. The experiment was designed to simulate conditions in the early days of our planet, and clearly showed that organic molecules that form the basis of all life can be made from basic inorganic building blocks. And a little spark. After Urey's death, it was shown that over 20 different amino acids were present in the mixture. An even more remarkable result than Urey and Miller claimed in their initial publication.
What are particle accelerators used for?
Particle accelerators are used to generate beams of particles moving at very high speeds, which are typically then collided with matter or other particles to learn about fundamental interactions. Most of the time the particles in question are subatomic particles, though atoms can also be used. Such experiments are used to address fundamental questions in physics surrounding the structure of matter and space. Typical modern particle accelerators are several kilometers long, with some operating in a linear fashion and others in a large ring.
Why is arsenic so poisonous?
Arsenic interrupts some of your body's most basic and common biochemical pathways. Arsenic, particularly as3+ and as5+ oxides, interferes with the citric acid cycle and respiration. specifically reduction of NAD and NADP synthesis biochemistry. If that weren't enough to kill you, and it probably is. Arsenic also boosts the level of hydrogen peroxide in your body, which causes another whole set of problems. Unfortunately, these toxic forms of arsenic are not only water-soluble but can be found in well water from natural sources and man-made contamination from mining.
What is a pyrophoric reagent?
Pyrophoric reagents are substances that will ignite spontaneously when exposed to air. Very often, this is due to a reaction with the water in the air. Thus pyrophoric reagents should be used only under inert atmospheres, such as in a glove box filled with an inert gas such as argon or nitrogen. Often, pyrophorics will be sold as solutions already dissolved in a solvent so that they do not tend to create fires so easily. Some of the more mild pyrophoric substances can be handled in the air, but caution needs to be exercised to flush air out of the container before storing them for an extended period of time. They also must be disposed of with caution, or they might accidentally set the waste bucket on fire. Perhaps not surprisingly, pyrophoric materials can be useful for starting fires in a controlled manner. Pyrophoric materials are present in the spark-generating mechanisms in lighters and some firearms.
What is alchemy?
Alchemy was among the earliest practices of a chemical science, and, in a way, it can be considered a predecessor to the modern science of chemistry. Alchemy is somewhat different than a modern science, though, in that it also has roots in mythology and spiritualism. Practitioners of alchemy were known as alchemists. Among the primary goals of alchemists were to find a method or material that could convert inexpensive metals into precious gold, as well as to find an elixir of life, which could make a person both youthful and immortal. Myths told of the existence of such materials and of the possibility of such feats. The goals of alchemists were based largely on these myths. In medieval times, alchemists could be found in many countries around the world, and those in different regions held somewhat different beliefs. In the Western world, people were still considering how to make metals into gold as recently as the late 1700s.
What is an electrochemical cell?
One common example of a redox reaction in electrochemistry involves the transfer of electrons from zinc Zn, metal to copper, Cu, ions in an electrochemical cell. Zinc, Zn, gives up electrons more readily than copper, Cu. So electrons spontaneously transfer from the Zn metal to the Cu metal. depositing Zn2 plus ions into the solution and causing Cu metal to come out of the solution and onto the solid. This process releases energy, which can be used to drive external processes, such as powering a light bulb, for example.
What are some different types of titrations?
We already considered the titration of a base by an acid. Certainly acids can also be titrated by bases. But there are also many other types of titrations, so let's look at just two more. Complexometric titrations. This type of titration involves using a titrant that forms a complex with the analyte. In this case, the species of unknown concentration is often a metal ion, and the indicator may be a dye molecule that forms a weak complex with the metal ions. The indicator ion complex is bound more weakly than the titrant ion complex, such that the titrant can displace the indicator from the metal ions. Thus, when the titrant has bound to a significant fraction of analyte molecules, the indicator molecules are displaced and the solution will change color. Redox titrations. A redox titration is a titration that involves the use of a reducing agent or an oxidizing agent to determine the concentration of the unknown species. In this case, the indicator can again be sensitive to the presence of excess titrant, giving a color change when the titrant becomes present in excess.
What's the difference between an H-bomb and an A-bomb?
The hydrogen bomb, H-bomb, is actually significantly more destructive than even an A-bomb. While A-bombs release energy via chain fission reactions, breaking apart heavy nuclei, H-bombs release energy through fusion of light nuclei. This energy comes from an overall increase in stability, due to the strong force that holds nuclei together as the light nuclei fuse to create heavier ones. To give an idea of the relative powers of these two weapons of mass destruction, consider that the A-bomb dropped on Hiroshima had a force on the order of 10 kilotons. Explosive force equivalent to 10,000 tons of TNT, while a common H-bomb has a force on the order of 10 megatons, or 1,000 times the explosive force of the A-bomb used at Hiroshima.
What is sugar?
A carbohydrate, a molecule composed of only carbon, hydrogen, and oxygen, that tastes sweet. Monosaccharides, or simple sugars, include fructose, galactose, and glucose.
What is condensation?
Conversion of a gas into a liquid.
What is precision?
The extent to which a measurement is repeatable. Note that this is distinct from accuracy.
Why did William Francis Gioke, United States, win the Nobel Prize for Chemistry?
For his contributions in the field of chemical thermodynamics, particularly concerning the behavior of substances at extremely low temperatures. Gioke's work did much to prove the third law of thermodynamics, originally proposed by Nernst, and helped make it possible to calculate the free energies of formation of molecules. Much of this was made possible by experiments performed at extremely low temperatures. And Gioke can be credited for developing the methods necessary to perform these experiments at temperatures close to absolute zero. Gioke's work explored the entropy, or disorder, associated with various forms of various substances. And he did much of the pioneering work in studying molecules at low temperatures.
What is the chelation?
Binding of a ligand to a metal atom through two or more positions.
What is thalidomide?
Thalidomide is a drug that was once used to treat the symptoms of morning sickness during pregnancy as well as to help with sleeping problems. A few years after it became widely used, people started to realize that thalidomide was causing birth defects in newborns. These birth defects included phocomelia, abnormal formation of limbs, facial features, nerves, and other parts of the body, problems with sight or hearing resulting from abnormalities in the eyes and ears, gastrointestinal disorders, Passli disorder of the face, underdeveloped lungs, and problems with the digestive tract, heart, and kidneys. The use of thalidomide was then discontinued. Though even today there is still some research underway into its possible use in treating cancers. Similar to the situation with DDT, the problems that occurred during the use of thalidomide were particularly influential in motivating the government to tighten regulations on testing drugs and pesticides before their use.
Why did Robert S. Mulliken, United States, win the Nobel Prize for Chemistry?
For his fundamental work concerning chemical bonds and the electronic structure of molecules by the molecular orbital method. Mulliken received the Nobel Prize in recognition of his work studying that nature of how electrons behaving in molecules. In particular for the molecular orbital approach that he developed. Molecular orbitals are formed by the overlap of the orbitals on individual atoms. and these can be used to rationalize whether bonds will exist between pairs of atoms. How strongly the pairs will be bonded, and what type of reactivity the molecule may be expected to undergo.
What is a ceramic?
An inorganic crystalline solid typically prepared by heating.
What is qualitative analysis?
Analysis of the identity of the constituent species present in a sample.
What is a dative bond?
A chemical bond in which one atom is essentially providing both electrons involved in the bond.
What gives a food its taste?
Molecules, of course. You probably learned in school that there are 4 basic tastes: sweet, bitter, sour, and salty. Your science textbook probably also had a diagram like the one on the following page, showing that your bitter taste buds are located on the back of your tongue, and sweet is tasted on the tip. Wrong. 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 Not only are there at least 5 basic tastes, but they're basically spread out evenly across your tongue. The 5 basic tastes are the 4 you know from elementary school. And umami. If you grew up eating Western cuisine, umami tastes like message, or maybe you just think Asian food. There is debate as to whether there is a 6th basic taste bud that senses fat. Or maybe another sensor for piquance, spiciness. But not even those 5, or 6, or 7. Basic tastes fully account for all the sensations you get during eating. Wine is probably the best example, if you drink wine, of course.
Does eating turkey make you sleepy?
You've probably heard that turkey contains a lot of tryptophan, which makes you sleepy. It's true there is tryptophan in turkey, but not more than is contained in most meats. It's also true that tryptophan is used to make serotonin, a neurotransmitter that makes humans sleepy. The problem with connecting these dots, I'm sleepy after Thanksgiving and tryptophan makes serotonin, which makes me sleepy, is that there are also a bunch of other amino acids in turkey. For amino acids to get into your brain, they need to use transporters to cross the barrier. Tryptophan is competing for a ride on the transporter molecules. With all of those other amino acids you just stuffed in your face.
What is the Charles's law?
a law stating that the volume and temperature of a gas are directly proportional.
Why are metals attracted to metal? Or why do magnets stick to your fridge?
Ferromagnets generate their own magnetic field. This means that other paramagnetic substances will be attracted to ferromagnets, just like they are attracted to magnetic fields. Most metals are paramagnetic, so magnets stick to your metal fridge. But your fridge itself is not magnetic.
Why did Alfred Werner, Switzerland, win the Nobel Prize for Chemistry?
For his work on the linkage of atoms in molecules, especially in inorganic chemistry. Werner was the first to propose the correct structure of inorganic compounds like Co, NH3, 4Cl2+. He proposed that the cobalt ion was in the center, surrounded by the ammonia and chloride ligands, in an octahedral arrangement. This proposal was consistent with the fact that two isomers of this compound were observed. As the two chloride ligands can be arranged either 180 degrees, trans, or 90 degrees, cis, apart from each other.